Hello students, welcome back to my channel BioPulse, and this is Chandrika. Today is our day 10 journey and we are moving towards the chapter plant growth and development. Today's chapter we are discussing about this, and this is the last chapter of botany portion of the first year syllabus. So I'm a bit happy about that because we finished 10 day for this series. That's what I'm a bit happy, and I will be full happy when we will finish all the syllabus, both botany and then zoology, isn't it? Yeah. Now, have you ever wondered how a tiny seed grows into a huge tree? Or how plants know when to grow, flower, or even sleep? Yes, of course, plants do sleep. In this video, we are going to make plants growth and development super simple and easy to remember, especially for your NEET preparation. So stay with me till the end because this chapter is much easier than you think. So let's begin. So for today, this is the first question from development. Question number 1. Development is option A, growth, option B, differentiation, option C, growth and differentiation, option D, growth minus differentiation, growth plus differentiation, and then D, growth minus differentiation. So here the right option is option C, growth plus differentiation is said to be what? Development. Next, question number 2. Growth of living being is or are A, irreversible, B, increase in size, C, increase in weight, D, all of this. Here the right option is D, all of this. Growth is irreversible. Once the growth is happened, we can't reverse the growth back, and it is increase in size. Of course, it is increase in weight also. So next question, that is third question. Swelling of piece of wood when placed in water is A, growth but not development, B, development but not growth, C, no growth and development, D, both growth and development. So here the right option is no growth and development. Plant growth generally is indeterminate. Plant's growth is generally what? Indeterminate. Fourth question. Plant grows A, for a limited time in life, B, for a very long time in life, C, for a very short time in life, D, for unlimited time throughout the life. So here the right option is D. Plants grow throughout their life. Not like us, okay, we grow only up to certain age, isn't it? But plants grow for unlimited time throughout the life. Fifth question, the given figure, this figure shows what? A, germination and development in beans, B, germination and development in maize, C, germination and development in pea, D, germination and development in grams. So here this is the bean seed, this one is bean seed. So germination, how it is going to germinate and then how it is going to be developed, that's one this figure is showing. Next question, question number 6. Plant grows throughout the life due to A, meristematic tissue, B, parenchyma tissue, C, epidermal tissue, D, more than one option is correct. So here the right option is A, meristematic tissue. Due to the presence of meristematic tissue, plant grow continuously throughout its life because meristematic tissue is present both in the root also and even in the tips of the shoot also, which are responsible for the growth and the development of the plant. Seventh question, open form of growth involves A, continuous division in all cells forming new set of cells which divide further necessarily, B. Division in some cells forming new set of cells which do not divide further necessarily. C. No division at all. D. Division in some cells at the time of injury which forms new set of cells to heal the injury. So here the right option is option A. It will continuous to division in all cells forming new set of cells which divide further necessarily. Wherever the necessary will be there what happens? Open form of growth will be there. 8th question. Secondary growth does occur in A. All angiosperms and no gymnosperms. B. Some angiosperms and gymnosperms. C. No angiosperms and only gymnosperms. D. All angiosperms and all gymnosperms. So here the right option is option B. 9th question. Secondary growth does not mean A. Increase in length of plant. B. Increase in girth of plant. C. Increase in diameter in plant. B. Both A and C. So here the right option is option A. Secondary growth doesn't mean that what length of the plant. See, this is the diagram which is given and then indicated as 1, 2. This is first one, this is 3 and this one is 2. Choose the correct label. What is first one? First one is obviously it is root area, means it will be root apical meristem because it is at the tip, and this portion is green and then it is shoot part, and if it is showing at the tip it will be obviously shoot apical meristem, and at the center what they will be presence of what? Vascular cambium. So here one is root apical meristem, second one is shoot apical meristem, and then third one is vascular cambium. So here the right option is root apical meristem is 1, and then second one is shoot apical meristem, third one is vascular cambium. So the right option is option B. Now next concept is about growth is measurable. 11th one. Growth can be measured by measuring increase in A. amount of protoplasm, B. dry weight, C. cell number, D. both B and C. Here the right option is both B and C. By the dry weight and then by the cell number we can measure the growth, how it is increased. Next one, 12th question. Match the column in respect with the measurement growth. Column 1 and then column 2. Maize, watermelon, pollen tube and then dorsiventral leaf. Here length, surface area, cell number and cell size. Watermelon, maize, how can you measure? You can measure by cell number, isn't it? And then watermelon by pollen tube by length. So C is 1. C is 1 means A, B, C. Dorsiventral leaf by surface area. D is 2. D is 2. C is 1. D is 2. Either it may be A or either it may be C. Among these two the answer will be right. And A is maize with respect to cell number, and then watermelon cell size. Maize is cell number and then watermelon is cell size. This is somewhat different question, isn't it? Yeah, but good question. We may measure the maize by cell number and watermelon by cell size, pollen tube by length and then dorsiventral leaf by surface area. So the right option is option A. Now questions from phases of growth. 13th one. The 3 phases of growth in correct order is A. Meristematic maturation elongation, elongation, meristematic maturation, meristematic elongation and maturation, elongation, maturation and meristematic. Keep in mind when 3 phases of growth is asked in order, always first meristematic cells will be there, the next they go elongation, the next they go maturation. So the right option here it is C. 14th one, cells in meristematic phase of growth, A have small nuclei, B have low plasmodesmatal connection, C have thick cell wall, D are rich in protoplasm. So here the right option is D are rich in protoplasm. Due to the rich in protoplasm, they will be very active. The meristematic phase cell will be very active. 15th question, the phase of maturation lies A proximal to cells undergoing elongation towards the tip, B proximal to cells undergoing elongation away from the tip, proximal to the cells of meristematic zone away from the tip, and then D proximal to cells of meristematic zone towards the tip. And then right option here it is B, proximal cells towards undergoing elongation away from the tip. 16th one, increased vacuolation is a characteristic of A meristematic phase, B maturation phase, C elongation phase, D all of this. Here the right option is option B, it is showing that maturation phase. 17th one, thickest cell wall present in meristematic phase, maturation phase, elongation phase, and both B and C. Here the right option is B, maturation. Thickest cell wall you can say maturation phase. Next question from growth rates. 18th one, increased growth per unit time is called A growth index, B growth rate, C growth efficiency index, and then D A and C. Here the right option is B, growth rate. Growth rate is nothing but increased growth per unit time. Add and then G is multiply. Here next growth rate is of two types. We can measure by two types, arithmetic growth and then geometric growth. Okay, arithmetic growth add constantly, there will be only addition, but in the geometric growth what happens, there will be multiplication, exponentially it is going to be multiplied. How you can remember, geometric growth will be J shaped and then logistic growth will be S shaped, sigmoid shape we can say, log, lag, and then stationary phase. You will all learn about those all things in the growth, isn't it? So here you just keep in mind about these two, arithmetic growth and then geometric growth. Arithmetic growth always add constantly, and whereas geometric phase always multiplies. Arithmetic A add and then geometric G multiply. So this is one of the example I am giving here. Identify the given figure. 19th question, identify the given figure. This one, A figure shows geometric growth with formula LT equals to L O plus RT, plus addition is there, means it is not. See here what happens, always the addition is going on, not the multiplication. Two cells are there and one more cell is added. Two cells are there already and one again two cells are added, isn't it? So like that, this one is wrong. And then B, figure shows geometric growth with formula W1 equals to W0 ERT. This is not the geometric one. Figure shows arithmetic growth with the formula LT equals to L0 plus RT. So here the right option is option C. If the figure is like this, this is representing arithmetic growth. Next, identify the given figure. See here what happens, it is going to multiply 2, 4 like that. 4 is again multiplying, again this is going to be multiplied, again this is going to be multiplied. If addition is there, it is said to be arithmetic. If multiplication is there, it is said to be geometric. A1. Figure shows geometric growth with formula LT equals to L naught plus RT. Plus is there, so this is wrong. Figure shows geometric growth with formula W1 equals to W0 ERT. So here the right option is option B. 21. Choose the correct option with respect to given figure of stages of embryo development. This is the stages of embryo development. First one is arithmetic growth phase. Second one is geometric growth phase. This one is geometric growth phase. He is saying no. 2 is arithmetic growth phase. No. And then 2 is geometric growth phase. Yes. 2 is actually geometric growth phase. 22. Choose a correct set of option for size or weight of organ against time. Against time. 1. W1 equals to W naught into ERT. LT equals to L naught plus RT. Linear growth curve, sigmoid growth curve, arithmetic growth, and then geometric growth. Here the right option is option C, 1, 4 and then 6. Correct set for size, weight of organ against the time. Next 23. The 3 phases of sigmoid growth occur in order as log, lag, stationary, log, stationary and then lag, lag, log, stationary, lag, stationary and then log. Here the right option is option C, lag, log, stationary. 24. In formula WR equals to W naught ERT is R is what? A. Related growth rate. B. Efficiency index. C. Ability of plant to produce new plant material. D. All of this. Here the right option is all of this. These all are representing the R. Relative growth curve, growth rate, efficiency index and so on. 25th. Here is the diagram. Choose the correct option. A. Absolute growth rate of A is more than that of B. This is B and then this is A. B. Absolute growth rate of B is more than that of A. More than that of A. Relative growth rate of A is more than that of B. And then D. Relative growth rate of B is more than that of A. Here the right option is option A itself is right. Absolute growth rate of A is more than that of B. See, absolute growth rate of A is more than that of B means why? Because see, initial it was 5 centimeter. Next, after time being, it is doubled. But here what happens? Initial it is only 50 and after the time being, it is just increasing the 5 centimeter. That's what. Conditions for growth. 26. The factors affecting growth can be A. Water, temperature, light and gravity. This one is the right option. 26. Option A is right. 27. About the differentiation, dedifferentiation and redifferentiation. 27. During differentiation, option A. Structural changes occur in nucleus and cytoplasm. B. Functional changes occurs in nucleus and cytoplasm. C. Structural changes occurs in cell wall and protoplasm. D. All of this. Here the right option is C. Structural changes occurs in cell wall and protoplasm during the differentiation. That's what they are going to differentiate it to specific kind of cells and then specific kind of organs. Dedifferentiation can be seen in the formation of intravesicular parenchyma. B. Intravesicular cambium only. C. Intervesicular cambium only. Intervesicular cambium only is the right option. 29. Read the following statements. 1. Cork cambium is a layer of meristem formed from parenchyma cells. 2. Intravesicular cambium is a layer of parenchyma cells formed from meristem. 3. Vascular cambium divide and produce cells that differentiate again. 4. Plant growth can be determinate or indeterminate. 5th, final structure of cells are never determined by location of the cell. 6th, the differentiation in plant cells are closed and dependent on the position. How many of the above statements are correct? And then here the right option is option A. 3 are correct among them. Next question from the development. Here the labels are given. This is 1, this is 2, and then this one is 3. Thirtieth question. Identify the correct labels. A. First one is expansion. 2. Plasmatic growth. No. 3. And 3 is maturation. 2 plasmatic growth, 3 is maturation. First one is expansion. So here first one is second one, this one is elongation, this is plasmatic growth, and then this one is maturation. So correct option is option B. Next, 31. Plasticity can be seen in A. Cotton, B. Coriander, C. Buttercup, D. All of this. Here the right option is D. All of this. Plasticity you can be seen in cotton, coriander, and buttercup. 32. Environmental heterophyly can be seen in larkspur, B. Cotton, C. Coriander, D. Buttercup. Here the right option is option B. Cotton. 33. The given figure shows this one, and this one. A. Developmental heterophyly in buttercup, environmental heterophyly in larkspur, environmental heterophyly in buttercup, and developmental heterophyly in larkspur. So this figure shows that this is the larkspur and developmental heterophyly in larkspur. 34th one. The figure shows sequence of the development process in A. In plant cell, in plant tissue, plant organs, all of this. Here the right option is option A. This is the plant cells which is showing the sequence of development process. Now the given figure, these two, is what? A. Developmental heterophyly in buttercup, environmental heterophyly in larkspur, environmental heterophyly in buttercup, developmental heterophyly in larkspur. This is not larkspur, so it will be among the buttercup only. So, but it is developmental heterophyly in buttercup. 36. Intrinsic factor responsible for growth and development includes intrinsic factors. Intrinsic factors means the factors which are affecting the growth of the development of the plant, which are present inside the body of the plant. Next, option A, intercellular plant growth regulators. More than one option is correct. Here the right option is more than one option is correct. Next is about plant growth regulator, that is PGR, and their characteristics. This portion is very important. Openly I am saying, because you will definitely 100% surely you will get one question from the PGR. Okay, so 37th question. PGRs are A. Small simple molecule of derives chemical composition. B. Large simple molecule of diverse chemical composition. C. Small complex molecule of diverse chemical composition. D. Large complex molecule of diverse chemical composition. So here the right option is A. They are very simple, where they are very small molecules of diverse chemical composition. So, trick to remember the hormones, that is PGRs, are AGCEB. Angry girl can easily bloom. Like me. Angry girl can easily bloom. Angry girl can easily bloom. Angry for Auxin, G for Gibberellin, C for Cytokinin, E for Ethylene, B for abscisic acid. From these hormones, these 5 hormones, you will definitely get 1 to 2 question. At least you will get 1 question, that is 100% pakkali sure. So next, let's see about the questions. 38th question. An example of adenine derivative PGR. Adenine derivative from the adenine who is going to be derived. Here the options IAA, kinetin, ABA and gibberellic acid. Here the right option is B, kinetin. Kinetin is a adenine derivative. 39th one. Gibberellic acid is A, indole compound, B, adenine compound, C, carotenoid derivative and then D, terpene derivative. Here the right option is D. 40th one. Abscisic acid is indole compound, adenine compound, carotenoid derivative and terpene derivative. Here the right option is C. Abscisic acid is a carotenoid derivative. Question number 41. Kinetin is a indole compound, B, adenine compound, C, carotenoid derivative and then D, terpene derivative. Here the right option is B, adenine compound. 42. A gaseous PGR is ABA, ethylene, GA3, IAA. You must not forget this one. The only gaseous PGR is ethylene. In your dreams also, you must not answer this question in the wrong form. So here the right option is ethylene. Ethylene is the gaseous hormone. 43. The PGRs that play important role in plant responses to wounds are A, Auxin, B, Cytokinin, C, Gibberellin and then D, abscisic acid. Here the right option is option D, abscisic acid. Next, 44. PGR ethylene can fit into A, group of plant growth promoter, group of plant growth inhibitor, B, C, both the group of promoter and inhibitors but mostly inhibitors, D, both the group of promoter and an inhibitor but mostly promoter. Here the right option is option C. It can fit for as a promoter also, as an inhibitor also, but maximally it is mostly for inhibitor. Next, discovery of PGRs. 45th question. Discovery of how many out of 5 major PGRs was accidental? Means what? Among the PGRs, how many are accidentally discovered, not purposefully discovered? Almost all, means 5 to 5, 5 out of 5 are accidentally discovered only. 46. Match the scientist with the plant they worked on. F.W. Went, column 1 with column 2. F.W. Went, E. Kurosawa, Charles Francis Darwin and F.S. Cook. F.W. Went is for oats, Kurosawa is for rice, Charles Francis Darwin is for grass, canary grass, and then F.S. Cook is for tobacco. So here the right option is option C. 47. Cousins confirmed the release of a volatile substance dash from the ripened dash that hastened the ripening of the stored unripened dash. 47th one, cousins confirm the release of a volatile substance dash ripened dash that has hastened the ripening of stored unripened dash. So here the right option is option D. 48, column 1 with column 2, PGRs are given and then which played role in their discovery. Auxin, Gibberellin, Cytokinin and then Ethylene. Here if you know any two of them, you can identify the right option very easily. Auxin is for avena sativa and then Gibberellin is for rice field and then rice one. Cytokinin is for tobacco, ethylene is for orange ripening story, isn't it? So here the right option is option B. 49th one, abscisic acid was independent discovered by 3 different researchers and named as A. Inhibitor A, abscission 3, dormant. B. Inhibitor C, abscission 2, dormant. Inhibitor B, abscission 2, dormant. D. Inhibitor B, abscission 2 and dormant. Here the right option is option C, inhibitor B, abscission 2 and dormant. 50th question, the term kinetin was given by A. Muller et al., B. Kurosawa et al., C. Cousins et al., D. None of these. The term kinetin was given by Muller et al. 51st question, the foolish seedling disease of a rice is caused by nematode, bacteria, fungus and virus. So here the right option is fungus. 52, Auxin was isolated by dash from dash. Francis Darwin, coleoptile of oat seedling. No. Francis Darwin, coleoptile of canary grass. F.W. Went, coleoptile of oats seedling. F.W. Went, coleoptile of canary grass. Here the right option is C, F.W. Went, coleoptile of oats seedling. The figure shows A. Experiment to show that tip of a coleoptile produce Gibberellin. Experiment to show that tip of coleorhiza produce Gibberellin. Experiment to show that tip of coleorhiza produce Auxin. Experiment to show that tip of coleoptile produce Auxin. Here the right option is option D, Auxin. 54, from the PGR, one of the most important PGR I can say. 54, Auxin was first isolated from A. xylem sap, phloem sap, human urine and root exudates. Here the right option is human urine. Auxin shortcut or else trick to remember, apical dominance. It will be helpful for apical dominance and adventitious root formation, and its main function is cell elongation, apical dominancy and root initiation. 55, synthetic auxins. A. IAA, B. IBA and then C. NAA. Here the right option is option D, both A and C. IAA and NAA are synthetic auxin. 56, auxin isolated from plant, that is option A, NAA, B, NIAA, C, 2,4-D, both A and B. Auxin isolated from the plants is BIAA. 57, auxins are generally produced by A. growing apex of root and shoot, epidermal tissue of shoots only, xylem secondary cells, endodermal cells of roots and shoot. Here the right option is option A. It will be generally produced by the apical growing apex of the root and then shoot. 58th one, assertion and reasoning. Auxin is used in plant propagation widely. Reason, auxin initiate rooting. Choose the correct option among them. So here the right option is option B. Both assertion and reasons are correct. 59. Auxin promote A. Rooting in stem cutting, B. Flowering in pineapple, C. Blotting in beet, and then D. Both A and B. So here the right option is both A and B. 60. Statement 1. Auxin prevent fruit and leaf drop at early stage. Statement 2. Auxin promote abscission of older mature leaves and fruits. Choose the appropriate option here. So here the right option is option C. Both the statements are correct. 61. Auxin A. It promotes apical dominance. B. Prevent apical dominance. C. Both promote and prevent apical dominance based on the condition. D. Can't say. So here the right option is A. It promotes apical dominance. Auxin always promotes apical dominance. 62. In tea plantation and hedge making, A. Lateral buds are removed. B. Shoot tips are removed. C. Root tips are removed. D. None of this. Here the right option is B. Shoot tips are removed in the tea plantation and hedge making. 63. Auxin A. Induces parthenocarpy. B. Act as herbicide. C. Help in cell division. D. All of this. Here the right option is all of this. Auxin help in inducing of parthenocarpy also, and it acts as a herbicide. 2,4-D is example for that, and then it is also helpful in the cell division. 64. 2,4-D is used to A. Kill gymnosperm usually. B. Kill dicot usually. C. Kill monocots usually. D. Both A and B. Here the right option is B. It uses unwanted dicots usually. Now next hormone is about gibberellins. PGR, gibberellin. 65. Gibberellins are A. Promotory PGRs. B. Inhibitory PGR. C. Neither promotory nor inhibitory. Both promotory and inhibitory. So here the right option is A. It's a promotory PGR. Gibberellin, G equals to giant growth. It will be helpful for the stem elongation, blotting break, and then seed dormancy. The main functions, the main effect of the gibberellins. 66. Which of the following statement are incorrect? A. There are more than 100 gibberellins reported. B. GA3 was one of the gibberellins to be discovered. C. All GA are basic. D. Gibberellic acids are also reported in fungi. So here the right option is all GA are basic. This is incorrect. Gibberellic acid, the name itself says it's in a acidic nature. Next, 67. Gibberellins is used in grapes for A. Increase in length of the stalk, increase in girth of the stalk, decrease in length of the stalk, decrease in girth of the stalk. So here the right option is option A. Increase in length of the stalk. 68. Gibberellins. A. Promote senescence. B. Delay senescence. C. Neither promotes not delay. D. Both promote and delay the senescence based on the situation. So here the right option is it delay senescence. Next question, question number 69. Assertion and reason. Assertion: spraying gibberellins on fruits extend its market period. Correct. Reason: gibberellins delay senescence. This is also correct. Just now we saw. Choose the best option. So here the right option is option A, because both are right and then reason is correct explanation of assertion. 70th one. Which of these are correct? Option A. ABA shows the malting process in the brewing industry. B. ABA spreads the malting process in the brewing industry. C. GA3 slows the malting process in brewing industry. D. GA3 speeds the malting process in brewing industry. So among them, the D1 is right. Question number 71. Gibberellin is used to improve yields of A. Apple, B. Grapes, C. Sugarcane, D. All of this. Here the right option is all of these. Assertion: juvenile conifers are sprayed with GA. Reason: GA delays senescence and malting period. Which one is given? Option are ready. Here assertion is correct but reason is wrong. So option C is right. Next question. Question number 73. Bolting is A. Yellowing of leaf, B. Node elongation prior to flowering, C. Early maturing and seed production, D. None of this. So here the right option is D. None of this. What actually the bolting is? Bolting is the premature production of flowering stem on a agricultural or horticultural crops. Prematuring production of flowering. 74. Gibberellic acid promote bolting in A. Beet, B. Cabbage, C. Plants with Rousselt habit, and then D. All of this. Here the right option is all of this. Next comes about the cytokinins. 75. Cytokinins were discovered as A. Kinin, B. Kinetin, C. Kinesin, and then D. Xantin. So it is discovered in as a kinetin form. Cytosine, C. Cell division plus cytoplasm keeper. It is helpful for the cell division and then delaying of the senescence. 76. Kinetin is a modified form of A. Purine guanine, pyrimidine cytosine, purine adenine, pyrimidine thymine, and it's a modified form of what? Purine adenine. Next question. 77. Kinetin was discovered from A. Autoclaved herring egg DNA, B. Human urine, C. Corn kernel, and then D. None of this. And then right option here it is none of this, because it is discovered from the autoclaved herring sperm DNA, not the egg DNA. So like this he will confuse you. Don't get confused there. Be confident on your concepts. 78. Read the following statements regarding cytokinin. A. Kinetin occurs naturally in plants. 2. Kinetin was discovered from coconut milk. 3. Zeatin doesn't occur naturally in plants. 4. Zeatin was isolated from the human DNA. How many of these statements are incorrect? Incorrect, he is asking. 4 are incorrect. 79. Naturally, cytokinin may be synthesized in A. Root apex, B. Developing shoot buds, C. Young fruits, D. More than one option is correct. Here the right option is D. More than one option is correct. 80th question. Cytokinin helps to produce A. Chloroplast in leaves, B. Elongation in sugarcane, C. Synchronized fruit set in pineapple, D. Flowering in pineapple. So here the right option is A. Chloroplast in leaves. Now next is about ethylene. This is one of the interesting and then neat favorite concept, I can say. Cytokinin and ethylene, especially kinetin, those all things. 81st question. Ethylene is synthesized in large amount by A. Ripening fruits, B. Tissues undergoing senescence, C. Newly developed leaves, D. More than one option is correct. Here the right option is more than one option is right. Ethylene it is ending hormone, we can say. Fruit ripening it will be helpful for, and senescence only it is helpful for the senescence, and it's a gaseous hormone. So among them these two are what? These two are right. 82th question. Ethylene causes dash growth of seedling, swelling of axis and apical hook formation in dash seedling. So here horizontal, vertical, monocot, dicot, monocot. It is useful in the dicot horizontal growth seedling. 83. Ethylene promotes A. Senescence and abscission of flowers. B. Senescence but not abscission of the flowers. C. Abscission of flowers but not the senescence. D. Neither senescence nor abscission. So here the right option is senescence and abscission of the flowers. Ethylene promotes. 84. Ethylene in fruits causes A. Rise in rate of respiration called respiratory anticlimatic. B. Rise in rate of respiration called respiratory climatic. C. Fall in rate of respiration called respiratory anticlimactic. And then D. Fall in rate of respiration called respiratory climactic. So here the right option is option B. Rise in rate of respiration called respiratory climactic. 85. Ethylene, option A, it promotes seed and bud dormancy. It promotes only seed dormancy. It promotes only bud dormancy, and then none of this. Here the right option is none of this. 86. Statement 1. Ethylene promotes internode or petiole elongation in deep water rice plants. Statement 2. Ethylene helps upper part of shoot to remain above the water. So choose the appropriate option among them. Here the statement 1 and statement 2 are incorrect. Sorry, they are correct. 87. Ethylene, option A, promotes root hair formation and increase in absorption surface. B. Demotes root hair formation and increase in absorption surface. C. Promotes root hair formation and decrease in absorption surface. D. Demotes root hair formation and decrease in absorption surface. So ethylene helpful for the promote root hair formation and increase in the absorption surface. 88. Ethylene initiates A. Flowering in pineapple. B. Flowering in mango. C. Synchronizing fruit set in pineapple. And then D. All of this. So here the right option is all of this. These are the initiated by ethylene. 89. Most widely used source of ethylene is ethylene, Haster's fruit ripening in tomato and apple. In aqueous solution is absorbed on the plant root. More than one option is correct. So here the right option is A. Ethylene. 90. Ethylene promotes A. Female flowers in cucumber increasing the yield. Female flowers in cucumber decreasing the yield. Male flowers in cucumber increasing the yield. Male flowers in cucumber decreasing the yield. So here the right option is option A. Female flowers in cucumber increasing the yield. Now about the abscisic acid, that is ABA we used to call. 91. ABA is A. Inhibitor of plant growth and metabolism. It's an inhibitor of plant growth but not the metabolism. It's an inhibitor of plant metabolism but not the growth. It's an inhibitor of neither plant growth nor metabolism. So here the right option is A. It's an inhibitor for plant growth and metabolism. So abscisic acid ABA, abscission plus bad conditions, and it will be helpful for the dormancy, and then stress hormone it is going to call, and then stomatal closure. It will be helpful for the stomatal closure. 92. ABA is a inhibit seed germination, promotes seed dormancy, inhibit seed dormancy, both A and B. Here the right option is both A and B. It inhibits seed germination and promotes seed dormancy. Inhibition means stopping. Promoting means it will initiate. 93. ABA is called stress hormone because it causes a stress to plant. It is released during stress in plant. It helps plant to fight stress condition. More than one option is correct. So here the more than one option is correct. 94th question. ABA stimulates A. Stomatal opening for more CO2 exchange. Stomatal closure to decrease transpiration. Stomatal opening to cause transpiration and cooling effect. D. Both A and C. Here the right option is B. For the stomatal closure to decrease the transpiration. 95. In most situations, ABA act as an antagonistic to Auxin, gibberellic acid, cytokinin and ethylene. It is in most of the time ABA acts as a antagonistic for GA. 96. Identify the correct label. This is 1, this is 2 and then this one is 3. Long day plant, day neutral and short day plant. So here the right option, this is the long day plant and this is the short day plant and the day neutral one. Right option is here C. How you will be remembering LSD plants? LSD plants means long day, short day and day neutral. '97. Read the given statement, statement 1 and then statement 2. Some plants require a periodic exposure to the light to include flowering. Statement 2. Some plants are able to measure the duration of exposure to light. Choose the correct statement. Here the correct statement is both the statements are correct and are about the photoperiodism. 98. For the day neutral plants, there is no correlation between the light duration and then flowering, direct correlation between the light duration and flowering, indirect correlation between the light duration and then flowering, direct correlation between the light intensity and then flowering. Here the right option is option A. It is no correlated between the light duration and then flowering in day neutral. 99. In photoperiodism, during of light period matters only. Duration of dark period matters only. C. Duration of light and dark period matters. D. Duration of light or dark period doesn't matter. Here the right option is C. Duration of light and dark period matters. Centuryth question. Question number 100. Photoperiods are perceived by A. Shoot apices, B. Flowering apices, C. Nodal buds and then D. Leaves. Here the right option is D. Leaves. Photoperiod are perceived by leaves. And this is the last question of today's chapter, plant growth and development, and even this is the last chapter of botany portion of the first year syllabus. So we finished 10 days of the series. Thank you for your support. Comment if you have any comment about my videos. Thank you. Bye bye.